they're giggling because I just said that. Multiple choice,
Oh,
fill in the blanks.
sorry.
Short answer, that's all right, it's worth repeating. Drawing, labeling, no long essay.
Sweet. Thank you.
It would be really interesting. I kind of get curious about long essay because it wouldn't take very much time at all to write the exam, but it'd take a long, long time to grade it.
What chapters do you think it's going to be on? Like,
Oh,
we're probably not going to get to everything. So.
Oh, it's going to be 3, 4, let's see here. Oh, crap. Let's just say it's not going to include chapters 7 and 8. Let's just decide that right now. Put that somewhere else. Otherwise, I'll have to hurry so fast that... So look at this. It will be the stuff about the viruses, biology of viruses, replication of viruses, cell biology of bacteria. There's a reading assignment on Moodle about the cell biology of archaea. I think there's a whole entire chapter about that, a whole bunch of it we could potentially put on there, but we're just doing some stuff about differentiation. There's also something about inclusion bodies that you guys are doing on your own, and maybe something else that's on there. But chapter 7 and 8 won't be on this exam. We'll put that on the next exam.
So is all that stuff up on Moodle then? Like the
I believe so.
questions?
Are the, oh,
There's
she's-
no 6 and 7. Yeah, there was, or 7.
there's no 6 and 7, and that's not going to be on this exam. Let's see what is on Moodle to make sure I know what you're asking me. Other announcements while I'm doing this. Remember the research presentations going on in the building today? Natural science students are doing posters, and humanities and social science students are doing short talks in 113. Let's see here. Chapter 1, independent learning, is right here. Chapter 3, select features. Oh. Maybe I put them on there. Let's see here. Oh, here we are. We're down here already.
Chapter 6.
Chapter 6 was up there.
The second one down.
There we go. Chapter 6 is here. So I'm going to put something up about archaea. It's not up there. So I'll be putting one more thing up. I'm going to do it as soon as I leave here today. Yep.
Is the lab stuff on the exam too, or is that?
It'll be on a separate exam at the end. It's going to relate, though. There will be days where what we talk about in lecture is related to what we talk about in lab, but there's not going to be content that's just in lab on the exam.
What was the reading you wanted for the inclusion bodies? Is that
Yeah,
okay?
I thought that was, let's see here, how did I screw that up? Let me see. Maybe I put it under my, I can make it show up right now. I switched to student role, so you didn't see all the garbage I have stored in here, because I didn't want you to freak out about all the stuff that could potentially be here. The reading is now listed right here. Chapter 3 and 4, independent learning. I think that's the one. Let's see. Come on. Yep, this is the one. So it's on Moodle now. Okay, other questions? Do we have any announcements about lab, Libby?
No, I'll be moving their plates later. They worked with differential and selective mean yesterday and did row cultures.
Wonderful.
And those two Winogronsky columns belong to somebody in this
Oh.
class. Yeah, there's some pink in here.
Awesome. So we're going to talk a little bit about the Winogronsky columns so you guys know what to look for, because you're making notes on these and describing them as they go. We're going to look at these all semester long, but this is a little schematic that shows some of the activities that might be going on in the tubes. Not every single tube is the same, of course, depending on where you got your soil.
And they will be, you all will be getting this
Yeah,
chart in a little while, okay?
you'll be getting this figure in lab. Yep, that's right. So it starts out in terms of a carbon source with cellulose, which was what? What did you guys put in there? There's probably dirt and dead bugs and
Sticks.
paper towels. Paper towels are made out of mostly cellulose. So we put that in there, and the first thing that happens is a couple of things, or the first things that happen. One is because you have it in a tube like this, the oxygen that might be down here gets used up as microbes consume any organic material that they can and use up the oxygen, which is their, for aerobes, or facultative aerobes, we'll talk about later, that's their preferred terminal electron acceptor. So they use that up right away. And then fungi chop up the cellulose, or bacteria that have exoenzymes. Remember how we were talking about transport the other day and how microbes can't do phagocytosis and they have to spit out exoenzymes that chop stuff up and then they can take in the monomers? The monomers can be taken up by bacteria that are in there, and if there's still oxygen left at that time, they can use oxygen as a terminal electron acceptor and use that up, or they can use it, and by it I mean sugar, right? The cellulose gets converted into sugar. Use that for fermentation and make organic acids. So what would be an example of, oh, here's an example. Acetate is an example of an organic acid. What other kinds of things would you get? Did you ever learn that anyplace? You could get formate, and butyrate, succinate, propionate, acetate. All of those things are substrates for microbes that are in the tube. So some of the microbes, and I know some of you guys have already seen this in your tubes, some of the microbes that are in there can use carbon dioxide and the organic acids to make methane. So when they do that, they're taking electrons from organic acids like acetate or other things, and they use CO2 as their terminal electron acceptor, which is a horrible terminal electron acceptor. That doesn't happen until the oxygen's used up and all the other things that could potentially be used are gone, and they make methane. And so in some of the tubes that you guys have upstairs, these don't have it, do they? That's all right. There's big cracks in the mud where methane got made and it pushed the soil out. And that would be quite a bit of force in order to make that much methane and move that mud. So that's what's going on there. We're going to talk more about that in the next unit. The organic acids are also substrates for a couple different groups of bacteria. The purple non-sulfur bacteria, which are my favorite bacteria. Let's see if you can see. This is asking for trouble. I can't see that at all, can you? You see the purple? You know what? I have a dream of making square test tubes. And you know what we could use them for? Winogrosky columns, because, never mind. Got to focus. Stay on task here. I can't see it. But this tube has purple patches in there, sort of maroon patches that are characteristic of purple non-sulfur bacteria. They can use organic acids as their terminal, or as their source of electrons, and get their energy from sunlight. And so the purple spots you see in there, and the different colors of maroon and purple and brownish, those are purple non-sulfur bacteria, phototropes. Also, the organic acids can be used by sulfate-reducing bacteria. And those are bacteria that use sulfate as a terminal electron acceptor. The terminal electron acceptor, instead of oxygen, so these are anaerobes, they can't use oxygen, so they use this as the terminal electron acceptor, and they generate sulfide. And there's evidence of sulfide production in some of these, and I'm assuming I won't be able to show it very well, but it just looks black. The reason you can see where sulfide production occurs is because if there's any iron in the soil, which is definitely in there, it's one of the most abundant elements in soil, that interacts with the sulfide to make a black precipitate, ferrous sulfate. So if you see black places in the soil that's otherwise brown, that is a place where sulfide is reacting with iron. The hydrogen sulfide can also serve as an electron source, a source of electrons for the green and purple sulfur bacteria. So you guys, some of you will see, and this tube already has places where rather than maroonish purple, it's got sort of chalky pink, and that chalkiness is due to the fact that they have sulfur in them. It makes them look chalky. And the purple sulfur bacteria take electrons from sulfide and generate sulfur, elemental sulfur, that they keep inside of their cells, and they carry it around with them as a terminal electron acceptor, or potentially as a source of electrons. They can get more electrons out of it here. The green ones, if you see green patches that have yellow around them, they're kind of yellowish green, those are green sulfur bacteria, and they spit out their elemental sulfur when they make it. That's what makes them yellow, those yellow patches. So they spit it out into the environment, which means that if they stay close by, they have access to it, but it's also available for the other organisms around them. So they're contributing to the community when they do that. And then some other organism can use their sulfur either as a terminal electron acceptor or as a source of electrons. So sulfur is in between, in terms of redox, in between sulfide and sulfate. It can serve as an electron acceptor and a source of electrons. We'll talk all about that later. Any questions about what to look for? And Libby's going to give you this sheet in lab, but now you'll kind of know how to relate what you see here to what you see in your tubes so that you know what's going on. I'm going to shove these up here before I make a big mess. Questions about this? More on this when we do physiology. So, bacterial physiology. Last time we were talking about cell components. When we ended, we were talking about the cell wall. And what would you say is a major, super important part of the cell wall? What is the monomer of the cell wall? Peptidoglycan. And you would draw it something like this. You're probably a better drawer than I am. These are the sugars, N-acetyluramic acid and N-acetylglucosamine. These are the amino acid, and what's the special thing about the amino acid? It's D amino acids, that's correct. Why is that significant? They're not susceptible. The peptides are not susceptible by proteases. So proteases typically cut between peptides, right? They don't eat the protein, but they cut between the peptides, and the D amino acids aren't susceptible to most of those. What were you going to say, Luke? You said same thing? And then these represent a couple of options. Either this line represents a covalent bond between these, or sometimes different bacteria put in a bunch of amino acids in there to make this whole big thing rigid. So we wrote down teichoic acids, but we didn't draw them in here. Teichoic acids are important for gram-positive bacteria because they link the peptidoglycan and the whole works to the cell wall. Here's a picture of what they look like in this diagram. There within the cell wall, the lipoteichoic acids bind the whole thing down to the plasma membrane so it stays on there tight. They have a couple of other important roles, and you can help yourself remember them if you look at the structure of teichoic acids. So the lipoteichoic acids and teichoic acids have a couple of roles, and one of them is sequestering magnesium ions. So magnesium is one of the elements that the bacteria need to grow. We'll talk about all of the ones they need to grow later. And the reason they're able to sequester so can you guess why they're able to sequester magnesium by looking at the structure? Magnesium ions.
Non-negative charge.
Yep, the negative charges, here, those negative charges attract the magnesium ions. It also, another important, let's see here. Well, these R groups actually are amino acids, chains of amino acids that are out here. But one of their roles is to sequester magnesium ions. They have a scaffolding role within the peptidoglycan. They link the peptidoglycan to the cell wall. So those are all important things that magnesium would fit right here. How many pluses are on magnesium? Two plus, right? Okay, so questions about gram-positive cell walls? Let's talk about, anybody? Let's talk about gram-negative cell walls. Where'd all my paper go? Yep, Ben?
Where exactly does the gram stain find in
The crystal violet?
complex? Yeah.
The crystal violet fits in between these subunits here. So the stain is small enough to fit in here. And it doesn't stay in there unless you treat it with iodine, which causes it to become insoluble and clump up. So that's what makes the gram-positives, that's why we call them positive, because they're positive for that staining. Okay, so what did I do with that giant stack of paper I just had? I think I'm cracking up. Why don't we do, Kayla, do you want to do your presentation while I get my crap together here? Oh, I know what the other thing I was going to tell you guys was. I put the sign-up sheet, this clipboard that you guys all signed up on, I put this on Moodle so that you can figure out when you signed up in case you forgot.
Okay, so my name is Kayla Knudsen, and I am going to do my presentation on dinoflagellates today. And so this started this summer. I studied abroad in Costa Rica, and it was a great experience, but my favorite part was going to, we went on a bioluminescent tour, and so we got to go see the bioluminescent water. And so this was the beach they took us to. We just went out on a boat. This is like an island here. And then after it got dark, we went out on a boat, went out, I don't know, 100 yards or so, and then we put on scuba gear and we got into the water. And as you were like swimming around and moving your arms, you can just see all of these little things glowing, and it's really pretty. Unfortunately, I don't have any pictures of it because we tried to take pictures, but they didn't turn out, and it all just looked black, so that was really sad. But they called it Las Estrellas del Mar, which is stars of the sea, and they call it that because it really looks like stars just in the water. And so this is kind of what it looked like. In the picture on the right, you can see it crashing up on the shore. We couldn't actually see that because we were there during a bad time of year. But we could see when we were in a boat like this, you could see all of the waves around the boat just were glowing the whole time we were on it. So it's super cool. And so bioluminescent is another picture of the waves on the beach. Bioluminescence is produced by a chemical reaction, which is in the presence of oxygen, and it's between a substrate called luciferin and an enzyme luciferase. However, this is a high energy cost for organisms that have this. There's a lot of bacteria that do bioluminescence. They do it for reasons of avoiding predators, camouflage, and courtship. And a lot of vertebrates will also use this. They'll have organs in their body that will house these bacteria, and that's how you get like fish and stuff that also glow. But dinoflagellates are the organisms that are most commonly the cause of the bioluminescence in the ocean that you can just see like this. And so they're a type of eukaryotic protist, and the most common genuses that have the bioluminescence are Ceratium and Protoparadinium. They have special organelles in their cells that are called scintillons, and that's where this reaction takes place. And it takes about 20 seconds for the reaction to take place, and it's in response to mechanical stress. And so when we're in the water, if you were still, you couldn't see the bacteria, or the dinoflagellates, but as soon as you like moved your arms or something, all of the water around your arms would glow. And so they were reacting to that movement, and that's why you can also see it on the shore sometimes as the wave crashes into the sand. So the purpose of the bioluminescence is not entirely known, but there are theories that it's a defense against predation because it disrupts the other organisms' swimming and feeding behaviors and kind of scares them away, basically. Cool. So my take-home points are bioluminescence results from a chemical reaction between a substrate called luciferin and an enzyme called luciferase, and dinoflagellates use bioluminescence as a defense against predation by starving and repelling predators.
Why do you suppose they only make light in response to the mechanical? What would be an advantage to that?
I think they said like as a predator would approach and like the water would move and stuff, it would make them light up and then scare away.
Or just startle them better?
Yeah.
I was thinking in terms of energy. I was betting it was because it costs so much ATP to make the light that they don't turn it on unless they need to. But
Yeah.
I like your answer.
Yeah. They also talked about how they have like circadian rhythms too, so like they won't light up during the day. You know, obviously you can't see it otherwise, but when
Yeah.
they've done it in the lab, they don't do it during the day either. It's only at night.
Any questions? All right, thanks, Kayla. Okay. Gram-negative bacteria. let's look at a figure of gram-negative bacteria. Write some stuff down about it. So we already talked about the gram stain. You guys are going to be doing it in lab coming up here soon. We're going to talk about the cell wall of the gram-negative bacteria. So this figure shows you the highlights. Here's the plasma membrane again down here. It's got transport proteins in it, which you guys have learned about before. Here is the peptidoglycan, the same molecule as what you would find in gram-positive bacteria, except for where's it vary? It varies between how the side chains get connected by interbridges, right? Some organisms have a direct covalent link and some have the chain of things in between. That would be one place that it varies. The big variability, though, is the amount of it. So in gram-negative bacteria, they have a thin peptidoglycan layer, very thin relative to the gram-positive bacteria, but they have an extra layer on the outside called the outer membrane. So the outer membrane covers a thin layer of peptidoglycan. I'm abbreviating peptidoglycan PG right there. Peptidoglycan. So the membrane is a little bit similar to the phospholipid bilayer, especially on one face. The inside face of it has phospholipids in here, but the outside, it's an asymmetrical bilayer. The outside has some different chemical characteristics and also some important things that are useful to the cell. So this is an asymmetrical bilayer. So this portion out here, the outside portion of it is a molecule known as LPS, or lipopolysaccharide. That is the outer layer of the outer membrane. So between the two of these things, it gives the bacterium a couple of advantages over the gram-positive bacteria. It creates a physical barrier, and what would the bacteria want to avoid out there? And remember that, and we'll learn about this more later, antibiotics that we use clinically are actually derived from naturally occurring products. And so the outer membrane actually protects the bacteria from antibiotics that are in the environment. So gram-positive bacteria, remember we said their peptidoglycan can be inhibited, the formation can be inhibited by a whole bunch of different antibiotics. The gram-negative bacteria have this layer on the outside that prevents that. So that's one of the important features. Another important feature is that it's involved in attachment to surfaces. So this out here, what's not shown is there are also a lot of proteins that are attached, but there's this provides some ability to attach to things as well. So similar to the, let's see, for some reason they don't show, similar to the lipoteichoic acids, there are also proteins that attach this outer membrane to the peptidoglycan. So remember when we talked about the teichoic acids and the lipoteichoic acids and the gram-positives? They attach the plasma membrane to the periplasm, or to the peptidoglycan. In gram-negative bacteria, they have these proteins that attach the outer membrane to the peptidoglycan. And for some reason, oh, here's the label. I just couldn't see it. Right here. Bronze lipoproteins. So, features, peptidoglycan, or lipopolysaccharide, we'll talk about more later. Bronze lipoproteins. The other thing that's shown out there that's really important for these guys are these. And again, that doesn't look like that is labeled. This is a big porin. Oh, here it is. It's on this one. It's hard to see the writing among the structures there. Porin proteins. So because they have a lipid bilayer around the outside, they have to have a way to get stuff across that, or they wouldn't be able to get anything through. It's great that antibiotics don't get through, but if they didn't have the porins, they wouldn't be able to get the stuff they need through. So they have porins on the surface that allows low molecular weight. Oops. weight solutes to get through. So what do we mean by low molecular weight solutes? That would be sugars. Sugars are small enough to get through. Amino acids, small peptides. They don't have active transporters out here to get them through, but they can just flow through this trimer. And those things have to get in because then they get down to here where they can be taken up by the transport system. So because this kind of cell, the gram-negative bacteria, have extra space in here and an extra layer, they have a bigger periplasmic space in the peptidoglycan. Or they have a big a big periplasmic space between the outer membrane and the plasma membrane. And again, there's enzymes in there that are involved in assembly, enzymes that are needed to get the outer membrane outside. So it's not just dead space. There's stuff going on out there that's important to synthesizing the cell wall. So let's talk more about this molecule that is involved in the outer surface of the lipid bilayer, the outer membrane. Lipopolysaccharide, which we always abbreviate LPS, lipopolysaccharide. Again, like peptidoglycan, you know what it's made out of based on the name. It's got lipids in there, right? Fatty acids and carbohydrates. So it's got sugars and lipids. So a few important things about it. There's a figure in your book that shows a little bit of a more detailed view. You don't need to memorize the structure of this. That would be ridiculous. But you can recognize it. And again, this is a schematic picture. It shows the lipids down here, fatty acid chains. They have some sugars here with phosphates in between, which makes this what? That would make it polar and hydrophobic. So this part's polar and hydrophobic. It's got a core of sugars that are less variable. Again, more hydrophobicity, phosphate. And then it's got an important part out here called the O antigen. So it doesn't tell us what sugars these are, and it shows an N rather than an actual number because it's variable among different organisms. And this is one of the ways that we can type organisms or classify them into categories. So there's a bunch of unique sugars out here that you wouldn't find in any kind of dietary situation, like abacose is one of them. I never even heard of that anyplace else. And they have different shapes, they have different branches, and they have different repeating units. And when this thing is actually present in the cell, it makes something with a lot of course, variable three-dimensional structure that is antigenic to the host that this organism might be in. That's why it's called O antigen. So the O antigen is the antigenic part. So lipopolysaccharide, it's got a couple parts. It's got one, the O, sometimes it's called the O side chain. O side chain, aka the O antigen. And just like this name tells you, it is antigenic. It's recognized by our immune system. So that is something that we can make an immune response against. So if you ever hear of O157H, have you ever heard of O157H7? It's a strain of E. coli. That's something you see in the newspaper sometimes. It's an especially nasty strain of E. coli that contaminates hamburger at fast food restaurants and spinach. And if you hear of deadly diarrhea that somebody got from food, it's oftentimes O157H7. The O in that stands for O side chain. And the 157 refers to strain number 157. They have them categorized that way. So that's a useful thing for us. Interestingly, though, although they're recognized by our immune system, some kinds of bacteria have the ability to change the antigenicity of their O side chain during the course of growth. So what does that mean? They must have the ability to synthesize different sugars or else put them together in different linkages so it changes the shape of the O antigen out here. So if you put different sugars in here, or put branches in different places, or put different numbers of them, you change the shape of this thing, which means it'll be recognized differently by our immune system, which is really great for them, bad for us, because then if you imagine you have an infection, you're making antibodies to fight it off, and then the bacteria switches to a different type, you have to start all over again. So it is antigenic, but it's antigenically variable. It can be varied by the bacteria.
that outer, this is the O antigen hydrophilic then, since it's on the very
Yep.
outside?
It's made out of sugars, so it's got all kinds of hydroxyl groups on there. It's hydrophilic and it helps associate water. From the perspective of the bacteria, if they're not associated with the host, that is one of the advantages of having all these sugars out here. It's not like they're going to eat the sugars, but they're a way to attract water to the cell. It's a good observation. Other questions about the O antigen? So that's one part of it. The other part of it that's interesting to humans and medically interesting is this part down here, the lipid A. So these are the lipids that are right here, sticking into this asymmetric bilayer. The book shows these O side chains as long, wispy things. Those are the O side chains. This is the lipid A sticking down there. And lipid A is important because it is toxic. It itself is toxic. In fact, it's known as endotoxin. So when people were first studying bacterial toxins, they recognized that there's some toxins that are associated with the cells and some that are not. And those that were associated with the cell were called endotoxins because people thought they were inside the cell. The others were named exotoxins. It turns out that there's not a huge amount of variability among the endotoxins in the world, but there's lots and lots of variability and specificity for exotoxins. And we'll talk about that later. So as the cells grow, imagine if this thing is growing or dying, and this is being exported out there, some of it sloughs off of the cell. If that gets into a person's bloodstream, that is sufficient to be toxic. And it can cause several different things, including shock, just from being, even if you have dead bacteria in you. Causes shock, fever, inflammation. Blood coagulation. We'll talk more about the mechanism of this a little bit. How many G's in coagulation? One. Thank you. Blood coagulation, hemorrhage, all kinds of nasty things. Have you ever noticed on medical supplies it says endotoxin tested? There's an assay. Have we ever talked about the Limulus polyphemus assay in here before, Libby? This is just coming out of my head today. I don't know what happened. But I think I must have got hit in the head really hard last night. But there's an assay that you can do that uses blood from a horseshoe crab. And point is here, when you have products that you're going to administer to a person, you have to make sure that there's no endotoxin in them. They might be sterile, and that's fine. But if they're sterile but have parts of dead bacteria in them, they can cause disease on their own. And so that's one of the things that products have to be tested for. And it's not really something the bacteria are doing actively. It's just part of the bacteria that gets into the host and introduces some things with interleukins and tumor necrosis factor that we'll talk about another time. Let's see here. This is some background about hypotonic. Do you guys have any questions about cell wall? There's some figures in here that are kind of redundant to things you've learned about cell biology. That figure about the hypotonicity has to do with what happens if the cell wall is weak. We talked about that on the first day and how the environment is hypotonic and that there would be a tendency to water to rush into the cell and cause the cells to explode. Okay, let's talk about some other features of prokaryotic cells that are located outside of the cell wall. Let's see here. Outside the cell wall. So almost all bacteria have a cell wall, or they have something take its place. But Tony?
Or we can talk about the inner part of, or the middle part of the alveoli.
The core polysaccharides?
Yeah.
Those are thought to be basically structural. They're sugars as well. They've got phosphates which make them charged and attract ions in water. But in terms of interesting functions, in terms of like human health and application, there's not as much to it. It's more about just extending this away from the cell, using these sugars as a structural component. Basically just something to label. And this book separates the inner core and the outer core. Some books just call it the core polysaccharides. Thanks. Okay, talking now about some things that would be considered optional equipment on a cell, on a prokaryotic cell. Things that they don't necessarily have to have. One of them is a capsule. So the capsule is a layer of material, usually composed of sugars, sometimes amino acids, that surrounds the outside of the cell wall. Whoops. Sugar or amino acid. This AA stands for amino acid. Outside the cell wall. So did you guys do capsule stains in cell biology? Do they still do that? Did you use Klebsiella? This is even the same organism you did. This shows a capsule stain. This bright area here is where the cell is present, and this pinkish area is the capsule. And in Klebsiella pneumonia, it's a big sugar capsule that's about as thick as the whole cell. So its volume is really, really huge, and it's a big, thick area around the cell. And it's protective to the bacteria and useful for a few reasons. One of the things is that it gives resistance, protection from phagocytosis. So it turns out that it's so important, its protection from phagocytosis is so important that the capsule is essential for the ability of some organisms to cause disease. For example, Streptococcus pneumoniae comes in two different forms, naturally occurring. One has a capsule, it's called the smooth form, because when it has a capsule on there, it looks all shiny and wet. The other one's called the rough form, and the rough one looks dry if it doesn't have the capsule. The rough form doesn't have a capsule. And only the smooth form is able to cause pneumonia. And actually, you guys might remember from genetics or someplace that when people were figuring out that DNA is the genetic material for cells and that it's important for carrying traits, the model they were using was Streptococcus pneumoniae, and the model had smooth cells with no capsule and rough cells with the capsule, or smooth cells with the capsule, rough cells without, and they found that if you take the DNA from the smooth cells and give them to the rough cells, they gained the capsule, which was really important for causing disease in a mouse model that they were using. So because this is a part or a product of a bacterium that's important for causing disease, what is the word that you'd associate with it? I think we talked about that on the first day.
Virulence?
Virulence factor, yep. It's a virulence factor. Whoops, not the phagocytosis, but the capsule. Capsule is the virulence factor. The part of the bacterium that's responsible for protecting phagocytosis is the virulence factor because that's important. The capsule is important for the ability of the cells to cause disease. So that's from our perspective. Why is the capsule important to the bacteria? So this is a giant layer of sugar out there. It's important for hydration. It attracts water. Sometimes they're associated with attachment. They're used for sticking to things. Either because another organism has a receptor that recognizes the sugars or the amino acids that are out there. Another thing that's important for the bacteria that have a capsule is that the capsule prevents attack by bacteriophage, which are what? Yep, bacterial viruses. So if they have the capsule on the outside, the virus can't associate with it and it can't get inside the cell. So lots of kinds of bacteria have capsules on the surface. That one happens to be a pathogen. The ones we talked about have been, but not all of them are. Okay, another important part of the cell are the pili. Pili, also known as fimbriae. Depending on what you're used to, I usually call them fimbriae. These are hair-like extensions of proteins on the surface of a cell. So let's see here. We're skipping over we're skipping over some stuff here. That's okay. Don't panic. This figure shows, and this is a electron micrograph that shows the fimbriae. You can barely see them out here. This particular kind of fimbriae are short little extensions. They're made out of proteins on the surface of the cell, and they're super important for attachment. Main role is attachment, and you can imagine that that makes them virulence factors for a lot of bacteria. So the book describes them as being hair-like proteins, but that doesn't really do them justice because they're assembled from a few different kinds of proteins. And on the surface of the pilus, at the very end, is an adhesant that's specific for the kind of surface it's going to stick to. So just like when we talked about viruses, remember there was a protein on the surface of the virus that had the role of binding to a receptor on the host? What was that on the virus? The spike proteins. This would be analogous to the spike proteins. So they're very specific for the target to which they bind. So sometimes the target that they bind isn't readily available. So if you have, I knew some people who studied fimbriae in, it doesn't matter what it was anymore, but the target for the fimbriae they studied were actually associated sort of right at the margin and in between the cells of our respiratory epithelium. And so the bacteria could only get to those sugars that were in there if the membrane is disrupted. So if you have, imagine the air is really dry during the wintertime and your respiratory tract gets all dried out and cracked, they could be exposed. Or if you're intubated and somebody puts a trach tube down your throat and yanks it back out, it gets all roughed up. When those surfaces get roughed up, they expose the sugars that are in here, but specific for the target to which they bind. For example, sugars. They get exposed, and then the bacteria have the ability to find a binding site and get a hold. So one of the main things they do is attachment. Another thing, in some cases, there are fimbriae that are used for a particular kind of motility called twitching motility. Next time we're going to talk about flagellar motility in swimming, but there's another kind of motility called twitching motility in which and another kind, in addition to that, gliding motility, I almost forgot. Twitching and gliding involve the use of fimbriae, the short little projections that kind of move back and forth using ATP. The cell uses ATP to move them back and forth, and that causes them to move around a little bit on a surface. It's not beautiful swimming like you get with flagella. Another kind is when they glide, they use them somehow. People are still trying to figure out how this works to glide across a surface. So gliding occurs on a surface. Both of these do. Let's see. These are on a surface as opposed to what we're going to talk about next time. Okay, the other kind of, so like I said before, fimbriae and pili are almost always interchangeable when you're talking about these kinds of activities. But there's one case in which pili and fimbriae are not interchangeable, and they're always called pili, and it's a totally different kind of structure. Not to be confused with the ones used for pathogenesis. That is the sex pili. Pili is plural, pilus is singular. So did you guys learn about conjugation in genetics? About how bacteria can mate with each other and pass plasmids or DNA between the two of them? I don't know if that's in genetics anymore. That's a process called conjugation. Or sometimes it's just called mating. So when bacteria mate, that's not where new baby bacteria come from. When bacteria mate, it's just used for the exchange of genetic material between two consenting adult bacteria, right? So it doesn't result in the production of new baby bacteria. But what happens is they form, and I'll show you a picture from the book. We're going to talk about this, as always, more detail later. But they form a bridge. This is on page 388 in your book. This cell has both regular fimbriae around the surface, short little ones, and it has this mating bridge, sex pilus. It says two E. coli stages in the early cells, in the early stage of conjugation. The F+ cell is to the right, This is the one that's, did you learn about the F factor? The F factor is the collection of genes that are required for mating and making this thing. So this cell has the capability of making the sex pili, and it makes them and extends them out, and it usually has just a few of them, maybe 10, but it's extending out in all directions, trying to link on to another cell of the appropriate mating type. And when it does that, it then deconstructs this thing. It sort of shortens, it contracts the bridge so that they're right up next to each other. I'll show you quickly. Here's the figure. We're going to talk about this stuff later. Here's the bridge. They get together, and then DNA from one cell can get passed into another cell, either part of its chromosome or part of a plasmid or the whole plasmid, depends on the situation. But it's really important in the environment because that's how bacteria can get new DNA, one of the ways. So it forms a channel. So this, rather than being just like a little tiny thin hair-like extension, it's actually more like a hollow tube or a channel. Forms a channel for DNA transfer. So not only can one E. coli swap DNA with another E. coli, when bacteria mate, they can do it with different species. They can swap DNA among other species, and so you can get a lot of mixing of genomes. If you can give part of your genome to another species, that organism that receives it can get a lot of important genetic information, like antibiotic resistance or new genes for a new biochemical pathway. All kinds of things can be transferred across. Again, more when we talk about that when we talk about genetics. Questions about fimbriae, pili, gram-negative cell walls? Pause a moment. We have 2 very good minutes and only 1 lecture before the exam. So look at your notes a second and think about what you might have questions about. I'm going to do the same to see if there's anything I want to reinforce. Next time we're going to talk about flagella, flagellar motility. We will, 2 of my favorite things from this chapter. Next time we'll talk about flagella and endospores. Luke, do you have your hand up? No? Did you have a question, Kyle?
The stuff we're talking about today is on the test.
Yes. The stuff we're talking about today is on the test. The stuff we're talking about Friday is on the test. What's not on the test is the stuff that I thought we were going to talk about Friday. So we're 1 day behind, but who cares, right? As long as we don't get 1 day ahead and I start screwing you guys over and making you do more than what you bargained for, no one ever complains. Tony? That is on the Moodle assignment I showed you. Yep. And I'll tell you what it's about. It's about the cell wall, a little bit about the cell wall and cell membranes of the archaea. And then we'll talk about other archaeal topics when we talk about physiology. Anyone? Don't forget to come to the thing at 3 today, poster session at 3. What else? What other special highlights are going on here? The inauguration is Friday afternoon.
See the faculty embarrass themselves in baseball on the 6th of Friday.
Oh,
That would be worth the price
that
of
will be worth
admission.
it. Okay. So those are the virus and the virally insensitive. So the ones that the virus needs to
Do you need those for the rest of the day? Yeah. If you need more, just ask me, and I remember they have like 50,000 from the activities.
Yeah, that's right.
I need to sell some.
Oh, for the virus, for the virus.
Alright, I'm gonna go home and make some food. I was planning on just eating here, but I realized I only took the Vyvanse this morning and not my antidepressant or anything. And like, I've never went without it, and I don't want to find out what happens if I do, you know? So,
But through the cell, then we can also
I'm
like.
gonna go do that, and then I'm gonna go to physics. So, I'll see you later.